It's time to synapse these chromosomes. Hey guys, today I'll illustrate and explain synapsis of a chromosome in meiosis using two examples. Crossing over is the exchange of genetic material between two sister chromatids during prophase 1 of meiosis that allows the mixing of parental and maternal alleles. This process leads to gametes carrying combinations of genes distinct from both parents and therefore responsible for genetic variation. So for individual 1, for the two chromosomes, we can clearly tell there's an inversion because the first two and last two alleles in the chromosome are inverted between the two. And we can tell it's a paracentric inversion because it doesn't involve the centromere. Now, to account for this inversion, we have to draw loops for our two chromatids for chromosome number 2 so that each of our chromosomes can be homologous in the tracks. So we draw our two bubbles and our two loops, and then we cross over the J and K loci because that is what the directions want us to do. They want us to cross over the J and K loci, which we do in between the two tracks. And then we just follow each chromatid track and find out the gamete types. So for number 1, this is where we check for aberrations in the chromosome to see if the gamete types are viable. So if there's aberrations such as deletion or duplication within the chromosome, then we know that the gamete is not viable. In number 1, we can see there's MLI centrome JKHG, and there's no deletion or duplication, so that means gamete 1 is viable. For gamete number 2, we can tell it's not viable because there's a duplication of the M and L allele in the chromosome. For number 3, we can tell it is normal and viable because there's no duplication or deletion. For number 4, we can tell that it is not viable because there is a duplication of G and H in the chromosome and it's deletion of certain. For individual 2, we have centrosome 1, which is MLI centromere JKHG, and then chromosome 2, GHKJ centromere ILM. So basically, there is no inversion or translocation in these two chromosomes. They're actually the exact same because chromosomes don't go right to left. They're actually the exact same if you just flip the second chromosome. So since they're the exact same, we do not have to draw a loop. We just have to draw normal bubbles, which I have. And basically we're just going to cross over from the J and K loci again, which I have. So now we did that, all we have to do is follow the tracks, and when we follow the tracks, we see that we relatively get the same chromosome for each gamete type. And all the gamete types are viable, which is MLI centrosome JKHG. So it's a 100%. So according to the information, there seems to be a higher level of expected fitness in individual number 2 than there is in number 1 because there is a higher variable of viable gametes. For watching, and until next time.